Hello, my name's David Wells. I'm a faculty member here at Texas A&M University at Galveston, and our lab is the Shark Biology and Fisheries Science Lab. And so our research is focused on fisheries science. Fish include both bony fishes, like tunas and trout, to sharks, elasmobranchs, sharks, stingrays, all different fishes throughout mainly marine ecosystems. So our research focuses on kind of three main areas. We're interested in movement and habitat use of fish. We're interested in what they eat, so feeding ecology, and we also work a lot on their life history. How long do they live, their age, their growth rates, their reproductive potentials. And all the work that we do is very applied, so we give this information to the funding agencies that sponsor the work, whether it's the government or state fishery managers, so that they can have that information to make rules and regulations and hopefully manage these species that we work on, so they're properly managed for long-term conservation. We've got a few different props up here that I can talk about specifically for some of our research. You can see some of the jaws from a short-fin Mako shark to a bull shark. This is a scalloped hammerhead cephalofoil, the big head part, and I don't know if you can see it closely. There's actually a little hole right here. That's a stingray barb. Hammerheads oftentimes like to eat stingrays, so oftentimes when you get a hammerhead and you look at close up at the hammer part, you can actually find barbs of stingrays embedded into the head, so that's pretty interesting. But so with respect to the movement, I know that some of the members of my lab have talked a little bit about the tools that we use. We have a couple different types of tags. These are both satellite tags that we use on different species, from sharks to other fish. One tag is what's called a spot tag, where we mount it on the dorsal fin of a shark, and when the shark breaks the surface up at the top of the ocean, this antenna will communicate to the satellites and tell us a very good location of where that animal is right at that particular time. And then when the animal goes back underwater, you can't receive any satellite technology, so we don't get any links. So the animal has to come up to the surface. So these are really good tags for real-time tracking of animal movement, learning where they're moving, migration patterns. This is also a satellite tag, but this particular tag is what's called a satellite archival pop-off tag. So the satellite, obviously, it links to the satellite, but it's an archival because it stores data inside the tag and stores it, rather than this one. This doesn't store any data, it just communicates your location. And so the difference with these tags is it also, not only does it get location, but it gets your depth and your temperature. You can program it second by second, minute by minute to take those recordings. And then at a pre-programmed time, this tag will sever off the animal. There's a little pin inside here and it'll burn, and this tag will then float up to the sea surface and begin transmitting all of the data to the satellite. So it's actually storing data during that whole period. Say you want it on the animal for 90 days or maybe a whole year, depending on how much information you want to gather. And then once that tag pops off the animal, floats up to the surface, it'll begin transmitting all the data up to the satellite, and then that information comes directly to us through our email. And we get all that information on where the animals moved. So those are a couple technologies that we use for some of the larger species, like some of the larger sharks that we work on, some of the hammerheads, the tiger sharks. blacktip, bull sharks, and some of the larger fish as well, mainly the tunas, yellowfin tuna, bluefin tuna, etc. But there's other ways to analyze movements of marine species, and it's called hard part chemistry. And so what we do is we actually take the hard parts, the vertebrae or the odolists, out of the fish, and we can analyze the chemistry within these hard parts and tell where they moved. So fish, bony fish, not sharks and rays, have these little bones, they're little ear bones. They're called odolists. And these odolists are really, really useful because we can cross-section them like a tree ring and count the number of rings so we can age the fish. In addition, we can analyze the chemistry between those rings and match it up to the chemistry of the seawater and try to determine where they were moving. Now, we don't get very fine scale location like satellite tag, but we get a general location. Did it move into the eastern Gulf, maybe the western Gulf, maybe down in the southern Gulf of Mexico? Or if a fish goes from freshwater to saltwater, we have some tracers that are very useful for looking at that transition. So those are really useful, but the odolists are only found in the bony fishes. Okay. Now, if you want to work on an elasmobranch like a shark or a stingray, you use the vertebrae. So we get the vertebrae from these animals, and we can do the same thing. There's rings that form, and those are annual rings as well. And so we can count the rings, just like the tree, just like the odolists, and determine the age of the animal. And we can also analyze the chemistry, again, between these rings to learn about their movements and migration patterns. So for those of you that think, I'm going to be a marine biologist, I don't need to study chemistry, well, look at here. Here we are, fisheries scientists working on sharks, and chemistry is a pretty powerful tool for us. This particular vertebrae is a really, really large one. This is actually a world record Mako shark that was caught off the coast of California, and they sent it to us to determine the age. It was somewhere around the 22 year age range, which is not the maximum age of this species, but it was certainly a very, very large Mako shark. So what got me interested in marine biology was just a general fascination for the ocean. I didn't really know exactly what it was about the ocean or marine biology that would get me excited. And then through college, I started learning different aspects of biology and marine biology, and I worked in a lot of different labs. And I worked in some labs that were enjoyable to work in, but I knew it wasn't something that I wanted to do, which was a really good experience, so that when I went on to graduate school and beyond, I kind of knew what I really wanted to focus on as a specialty when I ended up being more at the professional level. So I think one suggestion that I would have for entering students would be to get involved in research projects in different labs. First and foremost, get good grades, because when you go into graduate school, it can be competitive to get into graduate school as well if you want to move on and get maybe a master's degree or a PhD. So get good grades, work hard, but also realize that getting experience, just doing research, hands-on research in different labs is very valuable, even if it is just volunteering. It'll really help you learn what you like and what you don't like. And then for those of you that are really go-getters, and you really want to take this to do research on your own someday, we try to publish our information. That's another real important thing that we do, because if we don't publish our information, our results in scientific papers, nobody knows exactly what we did. right? The scientific community can't learn based on what we learned on a particular species. So I highly suggest anybody that really wants to really get involved in their own research to publish their own papers. How do you publish your own papers? You need to learn how to write. Writing is very, very important. So really try to get as much experience as you can writing. Take it seriously, because that's what we do a lot of. We're not out in the field all the time. We're not in the lab all the time. We spend a good chunk of our time writing papers, writing grant proposals, things like that.
So last question. So do you have a favorite fish or shark species, and why?
Okay, yes. So I would say my favorite fish are the bluefin tunas and shark, the Mako sharks. And they're similar because they can both regulate their internal body temperatures, which is not very common, right? They're endotherms, right, versus ectothermic. Most fish and sharks can't regulate their body temperature, so they're forced to move to different areas of the oceans because it gets too cold or it's too warm. They just like human beings, right? But Makos and bluefin tuna just have incredible migration patterns. I could go on for lecture after lecture telling you about their incredible life history and migration patterns. You can look that up for yourself for sure. But how do they do it is partly driven by the ability to regulate their internal temperatures so that they can go in areas that maybe a lot of other species can't do. So I would say those are two of my favorites.
Hi, my name is Emily Meese. I am a second-year PhD student here in Dr. Dave Wells' lab. I'm in the MARB IDP program. For my dissertation, I am doing a food web analysis of Matagorda Bay. So I get to sample basically every single trophic level that there is. So I sample a lot of primary producers. So how is energy getting into the system? And I have some samples here for that. So I'm sampling just water to get a particulate organic matter sample. I'm sampling benthic microalgae, so I'll scoop the mud and wait for the diatoms to rise up to collect them. And then I sample a lot of different vegetative producers, so seagrass, macroalgae, detritus, spartina, mangrove, anything that might be contributing energy to the system. I also get to sample all the different consumers, so any teleost fish or any elasmobranch, so a shark or a ray. I get to take a quick little mussel sample of them, and then I will bring that mussel back to the lab, clean it, and then send it off for isotopic analysis. So the goal of this research is to basically understand and quantify all the different levels of the food web using Matagorda Bay as a kind of a model ecosystem for a subtropical estuary to apply to other locations. And so what I do is I basically sample over different spatial scales and over time, and then we're hoping to kind of pull together, once you just have a mussel sample from a consumer, where is that consumer likely getting its nutrition from, and how is it kind of spending its time? So we'll be able to really understand the whole ecosystem a lot more when we're taking this bottom-up approach using the primary producers. So this research project is part of a really big grant, basically, where there's other professors from A&M at College Station and A&M Corpus Christi working together to kind of answer these questions about the Matagorda Bay ecosystem from all these different angles. So our puzzle piece was to do the food web analysis, and we may incorporate some movement of the upper predators as well. As a second-year PhD student, I still have a lot of options in front of me of what my goal would like to be. Ultimately, I think I'd really like to have a research-driven position. I do really enjoy teaching and mentoring, so I would like to incorporate that. I'm just not totally sure if I want to go the academic route now or not, so I'm still kind of testing those waters and seeing what I'd like to do. My advice to students that are just starting out in the marine biology or the marine science field would be to just get your hands on as much experience as you can, whether that's through the university or through your local aquarium or whatever is near you. Have those conversations with people that are in the field, learn what their day-to-day is like, and then get a hand on something. Even if it's something that you might not think you might like it, it's good to test that out to confirm which path that you really should go down.
Hi, my name is Mariah Livernoy. I am a third-year PhD student working with Dr. David Wells in his Shark Biology and Fisheries Science Lab. And I focus on the ecology and interactions between coastal predators, so large predatory fish species that live in the coastal zone on the Texas coast. And to study those organisms, I use a lot of different methodologies, but I will talk to you today about one of them, which is called acoustic telemetry. And this is a method that we use to monitor the movements and habitat use patterns of animals in real time. And how we do that is we use these acoustic tags. So these little tags have a coded signal that they produce. It's just a little noise, like a little ping, that they produce every about 60 seconds, and it's at a specific frequency and in a specific pattern. And these tags actually get implanted into the body cavity of a fish, so we surgically implant these tags inside the fish's body, and then the fish will swim around emitting the signal that's being produced by this tag. And as a fish swims past one of our listening stations, so we call these hydrophones. I don't have an example for you, but they're basically just a little piece of equipment that we put out in the water, and it listens for this specific tag. So it's the frequency and pattern of this tag. And when it detects that, it knows that a fish that has this tag implanted in it has swam past the listening station. So we have about 50 of those stations set up throughout Galveston Bay. So we can basically watch a fish move from one to the other of those stations using this internal tag. We have a couple of different tag types. These are obviously, one is larger than the other, but these are essentially the same tag. It's just that this one is bigger and can fit a bigger battery in it. So we use these tags on larger fish because they can kind of hold this tag, and it actually lasts up to 10 years, this one, but the smaller one only lasts about 4 years. So we kind of weigh our options of which kind of tag we use. We also put external tags on all the fish, so depending on the size and the type of fish, we use different tag types. So this tag right here is called a spaghetti tag. It's called that because it looks like spaghetti. And we put these using this sort of spear pole applicator. We attach this to the fish right on their sort of dorsal area, right below their dorsal fin. And this has a tag number on it, so we know which fish it is. But the point of these tags is really so that if a fisherman or someone catches one of our tagged fish that has our phone number and email and affiliation, like Texas A&M University, written on this tag, so they can actually call us and let us know that they captured our fish, and they can give us some information about where it was, how big it was, and all that stuff. So this is good for sort of a bony fish that's relatively large. For smaller fish, we don't want to put a really big tag because it might weigh them down and cause problems, so we use these little itty bitty tags. So this is a big stack of them, but each tag is just this small. And this is actually the same kind of tagging gun that they use to put clothing tags in, just the little T-bar tags that you get your clothing tags on. And these go into sort of small bony fish. And again, they have our phone number and affiliation so that a fisherman can call us and let us know that they caught our fish. And then for sharks, we use either the big spaghetti tag or we use what we call roto tags. So these are sort of a dart tag in and of itself, and I'll show you how the applicator works, but basically this goes around the dorsal fin of the shark, and then we clip it on. So then each side of this tag is sort of loose on either side of the dorsal fin, and it has an ID number and a phone number. And we use this funky applicator to put this tag on a shark. So we basically put one side of the tag and one side of this applicator on either side of the shark's dorsal fin, and then we clamp it shut like that, and then it's attached onto the dorsal fin. So those external tags are really great for helping us interact with fishermen and have fishermen interact with us, and it gives us a chance to understand what's happened to a fish after it gets captured by a fisherman, because there are a lot of people fishing in Galveston Bay, and we want to make sure that we interact with them and know what they're up to, and they can know what we're up to. What got me into fishery science, which is what I'm doing now, is I did my undergraduate degree at Northeastern University in Boston, and I was able to do a couple of different internships throughout, summers and different semesters that I was sort of off classes. So I got to try a lot of different things through internships and, research opportunities while I was an undergrad. And that really helped me decide on what I wanted to do ultimately in terms of my research focus, because I started working with phytoplankton and genetics, and I enjoyed it, but I was kind of, something was lacking. And then I did some outreach work, and, I really loved teaching, but I missed the research side of it. So then I moved into fisheries science, and I really love the interaction between people and the environment that we get when we study fish that are exploited by fisheries. So I really loved having lots of different experiences before sort of choosing the path of research that I was interested in. It helped me become a more well-rounded scientist. So I would recommend trying to get as many different experiences as you can, and just keep your mind open to different avenues of research that you might be able to follow. And it's really great to sort of interact with graduate students and researchers and faculty, by either working in their labs or volunteering in their labs, or even just talking to them. Your TAs are always available and want to talk to you, and will be happy to talk to you about research and their take on graduate school and things like that. So it's very helpful to talk to the people that are around you who are there to help you. I think my favorite fish right now, it changes all the time. My favorite fish right now is the alligator gar. It's one fish that I am working on for my dissertation. I'm studying their movements in Galveston Bay, because we have lots of alligator gar that live in Galveston Bay, the estuary, even though primarily alligator gar are freshwater fish. They live in rivers. So I am really interested to see if the alligator gar that we find in the estuary move up into the river, or if they sort of stay put in the estuary. Do they sort of join the others in their sort of normal habitat in the river, or do they basically stay in the estuary their whole life? So I'm using acoustic telemetry, which I talked to you about, to study their movement patterns, and right now they are definitely my favorite fish.
Hello everyone, my name is Travis Richards. I'm a postdoctoral researcher here in the Wells Fisheries lab, and I work in the marine biology department. And I'm going to talk with you today a little bit about what I'm doing as a postdoc here at TAMUG. And the major project I'm a part of is answering a very simple question. Right now, across the state of Texas, the popularity of fishing for sharks, mainly coastal species like bull sharks and blacktip sharks, is increasing exponentially. And one thing we're interested in knowing is whether or not these sharks, after they've been caught from the beach, survive. So typically, if you've seen somebody fishing from the beach, they catch the sharks, they drag them up onto the sand, they pull the hook out, they take some pictures, and they release them. And so both the fishermen and the scientists would like to know, how will these sharks do after they survive? And so similar to some of the other work we're doing in the lab, we're using electronic tags to answer these questions, but we're using a different type of tag. We're using what's called an accelerometer data logger, and it's this fancy tag right here, and it actually has several instruments all combined into one package. And so the big antenna, what you see here, is a VHF antenna, and we actually use this to find the tag after it's been deployed. We have a spot tag, which is just used as a GPS locator. And then inside the tag, we have the accelerometer, which records detailed information in all three axes. And so what's nice about this is, as the shark is released, it swims out with this accelerometer, and it gives us detailed information on how many tail beats per minute it's doing, whether or not it's listing side to side or dipping up and down. And so when we get this tag back after it deploys after 1 or 2 days, we can download all those data and see exactly how that shark is moving through the water, how long it takes to fully recover and go back to sort of a normal swimming pattern after it's been caught from the beach. And of course, we can tell by the accelerometer data whether or not the animal dies. So both survivorship information as well as behavioral information is recorded. All right, so the species that we're targeting are very similar to the species that we're targeting in other projects in the lab. Those are our major coastal species. So around here, it's a lot of blacktips and bull sharks. But opportunistically, people do catch and release scallop hammerheads. Here is sort of the skeletal prep from a hammerhead. So we'll be putting out these tags out on bulls, blacktips, scallop hammerheads, and a little bit further south, tiger sharks when we get them every once in a while. And the reason that we are interested in answering this question, whether or not these animals survive, is if you're managing any species or any population of animals, it's important to have an estimate of mortality rates, both natural mortality as well as sort of man-induced or human-induced mortality, so fishing mortality. And right now, the management models that people use for these species don't include an estimate of mortality from shore-based fishing. So by using these tags and looking at the behavior and the rate of survivorship of these species, we'll be able to answer that question and come up with a mortality estimate for the shore-based fishery. And that can sort of then in turn be used to inform the fishermen on best handling practices. If the mortality is really high, we can say, hey, things aren't working, we should handle the sharks a little bit differently, or try to do this to try to increase survivorship. Or if the survivorship is really high, and they're doing very well after capture, like, hey, you're doing a great job reviving these animals and they are mostly living after you capture. So in addition to talking about what I'm doing as a postdoc here in the lab, I wanted to talk to you a little bit about what I did as a PhD student, which was finished just last year. And I used methods that we use throughout the lab. I think you heard from Emily Meese talking about how we use stable isotopes to reconstruct food webs in marine systems. Well, I used that same method, stable isotope analysis, to reconstruct food webs in a unique environment, and that was the deep sea or deep pelagic environment in the Gulf of Mexico. And so the purpose of my PhD was to collect as many different organisms from the deep sea as possible, analyze the chemical signatures, the stable isotope signatures in their muscle tissues, and use that data to reconstruct the structure of the food web in the deep sea, because we know very little about the deep sea, and in particular, we know very little about food webs. And of course, food webs are crucial to understanding ecosystem function, because that is, that tells you how the energy flows through a system, from the primary producers all the way up through the apex predators. So if you want to manage an ecosystem, you have to understand how the food web is structured and how it functions, and maybe how it changes through time. And so I have here today some example organisms that we collected from the Gulf of Mexico, the deep sea environment, over the course of 4 years. And I just wanted to geek out a little bit about the deep sea and tell you about some of the really cool animals that we actually have here in our backyard. All right, here's a little sample platter of some of the species we get in the deep sea. And I pulled these animals in particular because they represent some of the dominant families of species that we have in the deep pelagic Gulf of Mexico. So I'm going to start over here in the top left with these two fish here. These are two different species that belong to the same family, and that family is Myctophidae, or the lanternfishes. And so lanternfishes are a group that are distributed worldwide. They are ubiquitous throughout the world's oceans. They are numerically one of the dominant groups of deep sea fishes. And that's important because they serve as an important sort of top-down influence in the food web, because they eat zooplankton constantly. So they swim up to the surface at night where zooplankton are plentiful. They leave the deep sea and swim up under the cover of darkness to avoid predation, and they feed heavily on zooplankton. And then they migrate back down to the deep ocean during the day to sort of hide from their predators. And while they're down there, they are often eaten by larger species. And by swimming up to the surface, feeding heavily, and then swimming back down, they're actually an important vector of energy from the surface of the ocean to the deep sea. So they actually connect species from the surface, the zooplankton, with species down in the deep sea who maybe don't migrate up. And the lanternfishes get their name because, like a lot of deep sea species, they can create their own light, right? They use bioluminescence. And what's interesting about them is they actually have these bioluminescent photophores along the sides of their body. That might be kind of hard for you to see on both species. And many of the species of myctophids have species-specific patterns of these photophores, and so scientists believe that they can use these to signal to their mates what species they are. Hey, I'm friendly, let's get together, let's mate. Because you got to think, if you're in the deep sea and it's dark and you're creating light, yes, you could be attracting prey, you could be attracting a mate, but you can also be alerting a predator to your whereabouts. So they have this species-specific sort of light signal that they send out. And then this species down here, which is called diaphis, is actually really interesting because if you look on the front of the face, you can see this sort of silvery section on its nose. That is actually a light-producing photophore. And so scientists think they actually use that almost like a searchlight to look for a specific prey using their own bioluminescence. So a very important and interesting group, the myctophids. Over here we have what is called a hatchetfish. You might have seen these featured on BBC's Blue Planet. It's another midwater fish. They also migrate vertically up through the water column at night to feed, but they have this nice silvery coloration that actually reflects the light around them and helps them blend in. And if you can look very carefully along the belly, it's kind of hard to see, they also have photophores. What's interesting about these photophores is they actually use these photophores to match the color of downwelling light to help them camouflage. So if you're a predator in the deep sea looking for prey, the way you look for prey is you get underneath them and you look up, because you're going to look for the silhouette that the fish's body makes because of the sunlight down filtering down through the water column. And if you want to get rid of that silhouette, you need to make your body the same color as that downwelling light. And so what's interesting about these hatchetfish is they actually have, it's hard to see, they have a photophore in their eye that they use as a reference point. So that photophore tries to match the color of the downwelling light. The eye sees that, and then that signals to the belly what color to sort of make the belly photophores. And that hides their profile from predators looking at them from below. So they have basically a way to eliminate their profile or their silhouette to predators looking from deeper waters beneath them. So hatchetfish, very interesting group. This species over here is pretty fascinating. This is a type of dragonfish. This is more of a predatory fish. These are smaller examples, but they get relatively large for deep pelagic fishes. They can get up to about 12 inches long. And they eat myctophids. They feed predominantly on myctophids. And you can again see they have sort of these light-producing photophores along their jaws or along their face. And like many deep sea predatory fishes, you can see how gigantic their gape is. Right? If I pull that jaw back, you can see it can open almost 180 degrees. And so they're very good at swallowing prey many times sort of their head size. And a lot of deep sea fishes have that ability. It allows them to sort of take advantage of any meal when it comes by. So this dragonfish is an interesting example of what a predator in the deep sea looks like. Again, very elongate, not great swimmers, mostly a sit and wait predator. Another sort of dragonfish-like species is this bristlemouth. Bristlemouths are one of the family is Gonostomatidae. They are perhaps the most abundant fishes on the planet. They're found in all the oceans of the world, and they are highly abundant. And this is an interesting fish because they have really nice examples of these photophores all along their bodies. And again, you can see these bristlemouths, which eat crustaceans like these shrimp, have pretty huge and wicked-looking mouths with lots of backwards recurved teeth to sort of hold on to their prey. So an important consumer in part because they are so abundant. Now we get to some of the crustaceans. Crustaceans, numerically speaking, often match the abundance of fishes in the deep sea. Interestingly, almost all of them are colored red. So these two migrate up through the water column at night to feed, but if you remember from sort of your intro to oceanography class, red light dissipates the fastest in water. So if you are red colored, there's no red light and you appear pitch black in the deep sea. And so they come in all shapes and sizes. These are two extremely abundant species. This is Notostomius in the deep sea, and we have a larger specimen here. But some deep sea shrimp, not this species but a similar species, they also use bioluminescence. They don't use it in the same way that the fishes do. They don't have photophores all the time. Some do, but this one species actually uses bioluminescence in a very interesting way. It's called a spewing shrimp, and when it's sort of startled by a predator, it will actually sort of vomit or throw up bioluminescent liquid. It will spray it out of its mouth, and that will either distract the predator, or some people say it will actually stick to the predator's face, and then that predator now has like this bioluminescent marker which could alert another predator to its presence, and that predator can come eat the other predator. How often that happens, we don't know, but regardless, they use the bioluminescent sort of vomit as a way to distract or deter predation in the deep sea. So just a few examples of some of the cool animals we have right here in the Gulf of Mexico, and there are many, many more to talk about.